Thank you for joining us. My name is Jermont Chen. I'm from the NIH, and this is our second SynBio and Control Theory breakout session. Our co-hosts today for today's meeting are Professor Domitilla Del Vecchio from MIT's Department of Mechanical Engineering, where she's the George N. Hatsopoulos Faculty Fellow in Interdisciplinary Research. Her research interests are control and dynamical systems theory and systems and synthetic biology. Our second also hosting is Tuba Fear from NIH's Eunice Kennedy Shriver National Institute of Child Health. Tuba directs programs in systems development biology and developmental genetics and genomics. With that, I'll pass the virtual microphone to our co-hosts, Domitilla and Tuba.
Hello, good afternoon, everyone. Thank you very much for joining for this meeting. I am Domitilla Del Vecchio, and here we have Tuba Fear. Tuba, would you like to say hi to folks?
Hi, everybody. So glad that you could all join us, and we're looking forward to an exciting and interactive session today. Please feel free to chime in anytime you feel like voicing your opinion.
Okay, so that said, I have about a 10-minute introduction to the session. So while people are continuing to join, I thought to spend this time just to give you a sense of what we would like to achieve with this breakout session. And to that end, I will give about a 5-minute background motivation. And then I will introduce the topic areas for discussion, about 3 minutes, and then the expected output, activity, and the schedule for the breakout session. And then before we start, I would like to go around the table so that you can introduce yourself, tell us why you're here, and what your research interests are. So that said, about 20 years ago, the field of synthetic biology was born. Probably most people joining this room know about that. The first two genetic circuits built were a bistable network and an oscillator. And at the very beginning of the field, people were building things to understand biology. But around 2010, there was a transition where people started to program biology for useful functionalities. And we have a lot of applications today, some of which have been already used in the field, such as cell-free virus detection for diagnostic. Then we have more futuristic applications such as targeted drug delivery, engineering microbiome, and then all those applications that interface with the human body and the human cell, such as engineered living materials and reprogram patient-specific cells. So with this beautiful vision, right, in 2006, people were proposing that we could create systems by composing simpler subsystems, right? So this simplifies design. We can forget the details that are inside the components so that we can create bigger systems just reasoning on input-output connections. And the reality is that in ECS, we do that all the time. We have been very successful, but feedback is often used to maintain input-output properties once we connect systems together. And the exciting fact is that if you look at historical trends of how quickly we can synthesize and sequence DNA, and the avenue of design automation software such as Cello and Comet has shown that increased scale is really becoming possible. So the trend is positive. But to some extent, this vision is been becoming difficult to achieve for a number of reasons. So first of all, we are still missing a way to compose subsystems such that we can achieve predictable design outcomes inside the cell. When components operate together in the cell, they often become unpredictable. And I think many of you that go to the lab, you do the experiment, it never works the first time, it never works the second time, and then you start debugging and teasing out piece by piece what's going wrong. Failures are very difficult to debug because they're often not due to direct interactions among the circuits that you build. They often go through interactions with the cells that you weren't expecting. Small changes in extracellular conditions, such as laboratory conditions or what's around your cell, can completely disrupt circuit function. We all know, for example, how sensitive our systems are to small temperature changes, small nutrient changes, things change a lot. So the strict analogy to computer engineering really fails in practice, right? In electrical systems, components are engineered to be insulated from external influences. through two main means. One is via physical separation, right? The current is flowing in a wire and it's isolated by everything else. And via negative feedback, at higher level, functions are insulated from external disturbances through feedback design, and we'll get more about it later. In biology, the components that we use are just not engineered for insulation. That's not the way biology built them, but oftentimes we use them thinking that they are insulated. So here just one slide overview of why parts are not insulated. So any genetic device that we build in the cell is going to have a behavior that depends on its context. Contexts include genetic context, such as the activity of surrounding genes, the directions of surrounding genes. And if you're working in mammalian cells, it depends on chromatin state. And we know well how difficult is compact silencing, for example, if we integrate circuits inside the genome. Cellular context is a challenge. Off-target interaction with cellular components, often very difficult to predict. Intermodule loading between your circuits, resource sharing. The cell, it's offering your circuits a large deal of resources for gene expression, protein modification, RNA modification, splicing factors, but we are using them and we are sharing between our circuits and the cell. Growth rate feedback, right? So anytime you engineer something, it's going to affect the way the cell grows, and that's in turn going to affect back your circuit. And then cell state, whether we are building our system in a fibroblast or in a pluripotent stem cell, the same circuit is going to behave extremely differently depending on the cell background. And then there are complications due to extracellular context, right? Competition for nutrients, right, with other strains and cells, conditions such as temperature and pH, cell-cell signaling, and if you're talking about more complicated instances, we also have the cell niche to consider. So anyways, I'm just going to give you one slide, a very simple example of how something that we take for granted, such as using the cellular resources, can make something fail miserably. So here is an example of an NGate. Is a system that takes two signaling molecules and gives you an output that should be high if both molecules are present. A very simple way to engineer that is to have two genetic activations, each one activated by the small molecule. If you combine them, model predicts, nothing should go wrong, both signals present, output is high. If you do a simple experiment without being careful and I specify what that means, you can get the exact opposite way. Okay, and that's really surprising, given that you're just adding these two very simple genetic circuits, and we are talking about creating enormous circuits. So what goes wrong in this case was actually not that difficult to detangle, but it's really due to the fact that you have to put your circuit back in the cell context. The cell is offering resources, in this case, RNA polymerase and ribosomes. Anytime the signaling molecule 1 is present, these resources are dragged in the first stage. There are none left in the second stage, so your NGate is going to fail. And here is an experiment of the model predicted through the standard Hill function model in dashed, and actually what the experiment gives you. So that's to say that robustness, okay, to variability in the cellular context is one big problem, okay? And here I have just one slide just to foster ideas on tools that we have from control system design. Control system design was born to ensure systems are robust to their environment. And the idea is that this way, if you insulate a module from the environment, you are hiding essentially the internal system complexities and just composing it as if it was insulated. So I want just to give you one very famous example, probably the first example where feedback control succeeded. And that's really probably the first instantiation of feedback controls. In the 1920s, when people were trying to build the first telephone line that would connect California with the East Coast. In that situation, people were having difficulty of signal attenuation along the line. So what people were doing, they were putting amplifiers in a series in order to amplify back the signals. But we know that amplifiers are very fragile to noise, to temperature changes, they have distortion, So bottom line, on the other end of the line, people were hearing junk. So this Mr. Black arrived and said, forget about designing vector amplifiers, just put a feedback around it. And the input-output response at this point becomes robust to all those uncertainties. So another example is the performance of amplifiers. Without feedback would be completely unrepeatable. Using the same amplifier in different applications will give you the different output. In any case, so I want now to introduce the two topic areas. So all this discussion is motivating the first topic area on capabilities of engineering biology. So the key question is, how much has the field of SynBio progressed since its inception in 2000, specifically regarding our ability to design behavior in a quantitative way? Okay, and this should be true despite all external influences that the field application, right, that we are envisioning will carry. And then the second point is, what can we learn from biology? I mean, biology doesn't have these problems. It's perfectly repeatable, robust, and accurate. So can we learn something by it? So the second topic area, it's about bottlenecks that we think are really the crucial steps to overcome to practical application. So what are the bottlenecks that make engineered cells usable for therapeutics or for basic discovery? And what can we use or reading food? Okay, so for sure, we know in engineering how to build complex systems. Aerospace, mechanical, automotive, electrical engineering provide all examples, right? We created the shuttle, right? We created a very complex systems every day, and luckily we are safe. So is there a common denominator, right, among all these applications that we can reuse or at least change to address these questions in engineering biology? So essentially here is the objective of the breakout session, to generate ideas for how engineering tools can be extended and in order to improve the current abilities in engineering biology and overcome bottlenecks. So we will have discussion, the scribe will summarize main points. I take all of that and create slides that I will present at the workshop on December 14th. And so now we can take a peek of how many people there are. And it looks like we probably are around the threshold value that we set in order to either split in two groups or stay in one group. So I would say that it's probably 24 participants. Is that about right? So then probably we can stay in one group. Tuba, yes.
Sounds good.
So then this is the schedule. So we will have 1 hour to discuss about topic area 1. Then we will have a break of 5 minutes. And then we will have another hour to discuss about topic area 2. And then the scribe will summarize and report back for the group. So with that, I would like to stop here for a moment before the round robin to see if I forgot anything. Okay, so then what I suggest we do, maybe we can go around the virtual room and we can each introduce ourselves, maybe telling us why you are in this workshop and what your research interests are. And then we can start with topic area 1 next. Okay, so probably I should pinpoint people. Jerome, do you have a suggestion of how to do that?
I was just going to probably read names off of the screen.
I will try, and I apologize in advance for my pronunciations of the names. Okay, so the first one in my order screen is Akos Nirjes, the Harvard.
Hi everyone, I'm from the Detected Evolution Genetic Code Engineering, and I'm here to learn about all the exciting topics we are covering and the activities in NIH in synthetic biology.
Thank you. Then we have Eric Johnson Chavarria.
Hi, everyone. I'm Eric Johnson Chavarria from NCI. I'm a program director of physical sciences oncology. So really looking at the intersections between synthetic biology and physical approaches perspective. So great to be here.
Then we have Mark Wilson.
Yes, I'm with a new startup bio company, Endure Biotherapeutics. We're based on these kinds of technologies. I'm just here to learn.
Then we have Julia Ringel.
Hi, my name is Julia Ringel. I'm a health specialist at NIH at the National Institute for Biomedical Imaging and Engineering. So I'm part of the group hosting these series of breakouts.
Thank you. Then we have Qing Xian Chang.
Hi, I'm a PhD student from ETH, and I'm interested in applying synthetic biology for medical applications. Great to be here.
Thank you. Then we have Ross Jones.
Hello, I'm Ross. I'm a postdoc, formerly was in Domitilla's lab.
Hi, everyone. Thanks for coming. So I'm Dave Rampula. I'm the director of the Division of Discovery Science and Technology at the Biomedical Imaging and Bioengineering Institute at the NIH. I've been involved in synthetic biology programs at the NIH for the last 6 odd years. And I'm here because I'm really excited to hear more about what engineering controls can do in biology, in particular the biomedical context. Being in the Engineering Institute at the NIH, I'm really excited to promote more engineering ideas around the agency to all my colleagues. So thank you all for coming, and in particular to my colleagues at the NIH for being a part of this. Thanks, Domitilla.
Thank you. Then we have Mitch Doctitz.
Hey, good afternoon, everyone. I'm Mitch Doctitz. I'm a staff scientist at the Oak Ridge National Labs, mostly involved in genomic science and analytical measurement tools, but I've had a long interest in synthetic biology and most recently more interested in synthetic community work. Thanks.
Thank you. Then we have Donghyun Lee.
Hi, everyone. My name is Donghyun. I'm a postdoc in Duke University with Ling Chong Yu in the lab. I'm using trying to study phase separation as a new tool for genetic circuit design. I'm here to study how continuous theory can be applicable for synthetic biology gene circuit design. Yeah, thank you.
Thank you. Then we have Anya Baitika.
Hi, my name is Anya Baitika. I'm an incoming assistant professor at Drexel University, and I'm interested in the design of biological feedback systems. I met Domitilla quite a few years ago. Hi.
Then we have Sebastian Palacios.
Yeah, hello everyone. I'm Sebastian. I'm a postdoctoral researcher at MIT Professor Del Vecchio's lab. I work in synthetic biology, but I'm obviously also very interested in advancing as a field the modeling and prediction of genetic circuit behavior. And I'm also really interested in the intersection of control theory and synthetic biology.
Thank you. Then we have Mark Wilson.
You already had me.
You had, sorry. Okay, so here things are mixing up. Then we have, let's see, Shukun Shi.
Hi, I'm Sukun Shi and from Vanderbilt, and I work with Professor Brown Jam, research associate professor in his lab. My background actually is circadian biology and sleep, which we have, as some of you probably know, we have transcription and translation feedback loop in that circuits. And right now, I'm interested in the synthetic biology and how this is involved in genetic control system. Thank you.
Thank you. Then we have Tai Seok Moon.
Hello, my name is Tae-Seok Moon from today representing Engineering Biology Research Consortium, the non-government or non-profit organization. I really glad because I see lots of young postdoc and student in this group. And I feel old, but at the same time, I feel young because of you. And I've been in synthetic biology almost 2 decades. Oh my goodness. I really interested in control theory and genetic circuit building for more than a decade, I guess. And I'm happy to see you, all of you.
Thank you. Then we have Asterios Arampatis.
Yeah, hi everyone. My name is Asterios Arampatis. I'm a PhD student at ETH Zurich. I'm interested in genetic engineering controller motifs in mammalian cells and exploring their use in cell therapy. Great being here and happy to learn more.
Thank you. Then we have Chang Wang.
Hi, I'm from NCI. I'm the branch chief of the molecular imaging branch. And the NCI is the extramural cancer imaging program. We're interested in intersecting molecular imaging and the SynBio approach, the tools for cancer diagnosis and therapy. Very much looking forward to learning lots of new information today. Thank you.
Thank you. And then we have Thorsten Schläger.
Hi, sorry, I was muted. Yes, my lab mostly works on pluripotent stem cells. I'm mostly here to learn. I'm not sure if I can contribute all that much. We have been collaborating with Domatilla's lab for a while on reprogramming, but I'm really here to learn.
Okay, thank you, everyone. I hope I haven't missed anyone. So if I did, please raise your hand. Okay, good. So I think with that, we can probably get started with the discussion. So in particular, we should start with topic area 1. So we have about an hour for that. So capabilities of engineering biology. So I think here really the key questions is how much has the field of SynBio progressed since 2000 regarding the ability to quantitatively predict circuit behavior as opposed to just qualitative prediction? And to what extent are we able to engineer a system that behaves as intended in defined conditions? So at least in the lab where everything in the environment is well controlled. And instead, to what extent can we engineer a system that behaves as intended despite the conditions are changing around it? For example, in the field, or in a reference cell line versus a true cell line, for example. And then to what extent we can learn from biology to design more robust, repeatable, and accurate systems in the face of uncertainty. And here we have a plethora of examples from biology regarding robustness, repeatability, and accuracy, right? For example, the timing of signals in the embryo development is accurate, precise, and repeatable. Why can't we achieve the same with the same pieces, essentially? And can we learn from natural process of evolution in order to achieve these goals? So for that, I will just open the table here for any comments. I will please raise your hand or just talk in or put your thoughts in the chat.
May I go first?
Yes, please.
So, I mean, I've been here in this field, almost 20 years. And, to me, synthetic biology tremendously, achieved a lot last 20 years in terms of the, prediction and quantitative, analysis and then building. But I probably need to talk some bottleneck, next topic, I guess. But in terms of, defined condition and then prediction, including, some example like Chris Boyd's amazing circuit published in Science of a few years ago, we now have ability to, predictably build and, analyze, genetic circuit. Still, we still need to consider, of course, host effect, how to transfer that knowledge into non-model organism other than E. coli and yeast. But that's probably the topic for the next hours, I guess. So I will stop here.
Thank you very much. Any comments picking up on that? So here I'm going to just pick up on that. So there are great examples, right? We can achieve quantitative prediction of composing circuit in defined lab conditions, right? Your bacteria growing in exponential phase at that exact temperature. What's the ability to extend those behaviors to more realistic situations, or even in a situation where you just don't know what's going to happen in the environment? Any thoughts on that? Or for the more biology-oriented people, why can biology do this computations and signaling timing so accurately while we are trying to use the same pieces and we can't quite as well?
Because we don't know enough about biology. So I'm struggling a little bit because, you know, your very first question here about the field of synthetic biology since 2000, you know, biology has progressed enormously in the last 20 years. And even the word or phrase synthetic biology, I'm not sure what, if you're trying to limit its definition to gene circuits, let me know now. Because it's not limited to gene circuits in any way. But gene circuits very much is related to metabolic engineering, which has been around even longer. And they all have the same problems. And it's until we understand what the side reactions are, what the promiscuity is, what the specificity is, it's always a struggle. I can go on and on, but don't mean to.
So the point is that we don't know enough about how what we build is going to interact with its surroundings?
I think you need to define its surroundings, if it's a molecule within a cell or a cell within its environment or within a tissue. I mostly work in the area of microbes and bacteria.
Mm-hmm.
I do a lot of cell-free work where we try to reduce that environmental complexity, as well as component complexity.
So how do we reduce environmental complexity? Just removing things?
In cell-free systems, we pull proteins out all the time. Of course, people do knockouts in live cells all the time as well. There's a lot of ways to do that. I don't mean to sound pedantic. It's just I'm not sure what you're asking. Do you want explicit examples of synthetic biology progress?
So no, I'm just wondering, so there is a big difference, right, between cell-free and in the cell, right? So in cell-free, somehow you have control on all the components that you have in the system. Yet even there, you have resources that are finite, right? So you start your sensor, it goes up, and then at some point you run out, right, of polymerases and things of that sort. So there are other types of issues. So my point is probably more than, so I think cell-free has its own issues, probably in cell being bacterial and mammalian have separate types of issues. It's more of a question, mine, of what type of issues, right, do you see related to the environment in each one of this, and what is the environment, right, of your gene circuit or your gene expression module in each one of these setups?
No arguments on any of these. I think every host or chassis system you use has its own features, some advantageous, some not. And each one defines a different environment or a different level of control. We all make flow systems, you know, we all have ways to get around some problems, but not all of them.
So one of the questions I was thinking about is when, you know, everyone says, even in the last breakout, we talked about systems biology and not knowing enough. My question is, when do we know enough? Because we could do systems biology forever and not do synthetic biology. At what point do we say we have enough? I guess it's you just conceive an experiment and go from there, because then you learn from that. Then you start to learn more about the system, and then you can engineer what you need to.
Absolutely. I don't know when we know enough, but I know that we skip things all the time. Like I said, we've been doing a lot in the lab with synthetic microbial communities. And so I'm just jumping over molecular networks, not necessarily, but you could work on that scale and learn a lot. And so we try to mix whole cell metabolic models with multicellular systems. And we don't know enough on either scale to do it well.
So can I comment on point or some other bit unrelated point? So I mean, to me, biology is very complex and messy, as you know, but still, not other physical system also very complex as well. If you looking back, the history of the physics and chemistry. But one issue in biology, of course, would be, I would say, uniqueness of individuals. And every single species, or every single human, or every single bacterium different from another. That is kind of one challenges in biology in general. And I keep asking myself, what would be governing rules in biology and synthetic biology? Do we have any governing rule other than evolution? And then because evolution also affect, synthetic circuit behavior, at the same time, characteristic of the, whatever circuit and biology itself. And because of that, there is no generalizable rules that could be applicable to all the biological system. So complexity is one issue, but to me, I mean, the more fundamental issue is we don't have those kind of governing rules that kind of applicable to all the system in biology. That's kind of my personal opinion, I guess.
I would add to that too. I wholly agree with what you just said. It's not just that, we don't have fundamental laws for biological systems,
right,
like the physical sciences. And I find it outrageous that we could design chemical plants because we understand thermodynamics, but we're going to jump ahead and engineer biological systems without having any idea about stochastic limits, noise, how do we deal with that, scaling relationships, feedback laws, rules, all the things that we know go on, but we don't have any basic laws.
Yeah, so but another point I want to make is, despite the lack of governing rules, we are not necessarily need to understand entire biological system for the real application at the end of the day, because I mean the application does not require we kind of finding whatever entire governing rules. So that's why I'm still optimistic in synthetic biology, because we could kind of make the small device and system that could be eventually lead to whatever application we need, from the NIH perspective.
So we said that there are no fundamental rules, right? No basic laws like F equal ma that we have, right, for physics. So what about, and here's just a thought, but what about if we go back at the molecular interactions and we start writing out in extreme detail, right, all the thermodynamics and the force interactions among molecules? If we go down at that level, there are fundamental laws, I will argue. It's just that probably we can't parse them. They're too complex once we put everything together. So I can see there are fundamental laws. So, but
chemistry is still chemistry. I don't think that changes, but we don't have the protein binding characteristics for protein A and 10,000 other proteins. For example, we know central metabolism fairly well. The enzymes of central metabolism have KMs and turnover rates that are wholly different than the rest of metabolism. The substrate of a kinase could be dozens of items, but it probably prefers one or two more than the rest. And I mean, I know we've seen this. We've done these experiments. We've tried to recreate these systems and pathways, and that's exactly what turns out.
Yeah, I mean, Domitila, I mean, I understand your point, but to me, those thermodynamics law and whatever other law, I mean, applicable to biology, that is the physics law or chemistry law rather than biology law. We have the biology law that is the basically central dogma, of course, and then also evolution. That's for sure. That is the governing, rules or principle applicable to any single organism in planet. But other than that, what do we have to apply when it comes to building generic circuit and others? I mean, we have some small guidelines, or I would say, I don't know how to say that one, but some guideline we can use. But other than that, do we have any generalizable rules that could be applicable to every single organism in the planet? That is kind of my struggle. And I try to ask that question every day, but I still don't know the answer. If somebody answered that question, I believe that is the Nobel Prize, of course.
So then if we feel that the issue is much more fundamental to the point that we don't have governing rules, to what extent it makes sense to ask, can we just predict circuit behavior, right? So can we predict how the system is going to behave in such and such condition? If we don't have fundamental rules, how can we address the prediction or the design question which requires prediction?
I mean, individual kind of characteristic, we kind of define whatever rule within that specific strain or species. If we admit that limitation, we can make some rule. But if you transfer that knowledge to another organism, probably that rule doesn't apply. So we have the rules, I mean, so that's sufficient enough for some application. But if you're thinking about governing rule, we don't have. But still, that's okay. We don't need that kind of governing rule for applicable to entire organism. That's my point. That's why I'm optimistic still.
So Tuba, are you monitoring the chat? I saw a few people entered comments in the chat.
I am monitoring. I responded to a couple people, and I would like to invite them to actually talk a little bit more about what they wrote in the chat, Ross and Shuku, and if one of you or both of you could comment on what you've put down in the chat. I thought it was really interesting.
Yes, thanks. I don't know much about other things. I think about the one thing is in the biology, during the evolution, there is so complex there. However, in the biology, when we investigate the questions, we use two tools. One is overexpression, one is loss of function, another is gain of function. So for the disease, I think, I know a little bit about disease perspective. So if we know the cause of the molecular cause of the disease, so then we will have the target. So then either loss of function or gain of function. So then the engineer or synthetic biology will play a role here. So the great things here for us is first we know the target. Secondly is we do the therapeutic things. One very great question is, a lot of you have already pointed out, is how we sense that in a same system we sense the problem. Then we do the therapeutic approaches. So then how we respond to this molecular cause, then if we can detect that cause very early, so then we do the do the therapeutic response. So will that helpful or not? Will that be interesting or not? So I think it not only just separate the two things. One is to find the cause. Another is to do the therapeutic effects. So how we, when the disease or this change or dynamic problem happen, when the mutation happens, we detect that. So then we use this as an engineer target and to do the treatment at the same time, as long as we find the problems, find the molecular mutations or this kind of thing. How can we connect this together? I think this is connected to the circuits and just now, a lot of you have already talked about that. Not to separate these two steps, and however is connect these two steps, find the problem, so then do the therapeutic approaches.
I do have a question actually for the synthetic biologists out there. What I'm hearing from the discussions, especially earlier discussions, is that there is an issue of the issue of scalability in that, what's the scale of the space that you're trying to control through the use of these synthetic biology tools, right? So because I'm a, biologist, so I look at, and somebody mentioned, networks in biology are very complex and you need to think of it in a systems level. But, when you think of it in a systems level, there are different, levels you can go up to. Like, what's the space? Do you start out with the size of the space you're trying to control, or you just start with the very basics, molecular, down to the chemical reaction aspect of things and try to build it out? If you do that, you'll run into more problems with trying to replicate what you're trying to replicate because you're starting too small without taking into account the bigger picture. You don't have to think about the entire, say, embryo, but you can think of the space within the cell that you're trying to control, or the area of the tissue you're trying to control. Am I making sense here? Like, do synthetic biologists have that scale in mind when designing their tools?
Anyone?
Well, we can fight on this a little bit. I think definitely the classic SynBio approach, at least for the circuit development side, is let me make a circuit and let me focus on that specific thing. And then you run into the issues of the cell because it turns out that you're way overexpressing proteins or whatever it is that you're doing. And so then you have to think about what is this context, what is the space that is engineerable within the cell. But I do think that on the flip side, some of the applications of the work with, for example, engineering cell therapies or things like that, you are kind of starting with the context of what the cell is trying to do already, and then you're trying to sort of engineer the cell within those confines. And so maybe people that have been working in that are still approaching it from the tool development angle of, let me just make this thing that does X in the cell. But I could imagine maybe with some of these applications, it would be sort of easier to integrate like the picture of the cell from the beginning. But I mean, from the perspective of the cell, I think something that I wanted to add was that it's not like these problems of robustness and so on don't exist. So for example, resource competition and things like that do exist within the confines of how a cell will normally operate. And you can see redistribution of the cell resources between different genes when they respond to different sort of inputs in their environment. But the cell has sort of evolved to either take advantage of that on purpose. So sometimes that redistribution of resources becomes a useful way for them to globally regulate their gene expression. And sometimes it's a way for them to acutely and strongly control one gene transiently, but then they turn it off quickly so that it doesn't have too long of a detrimental effect. And so I think that if we sort of look at how the cell deals with some of these things, then we can maybe integrate that into how we design systems. And so maybe that gets to sort of building within the confines of the cell that you're kind of getting at too, but I'm not sure what anybody else thinks about that.
I agree with you. I could rephrase maybe some of what you said is I don't think we understand or we always know what the cell's objective is versus our engineering objective. That comes back around to understanding, I'm not an engineer, but the cell's budget for like noise, I said, another budget is energy, right? Another budget is resources, carbon materials, nitrogen, et cetera. And of course, you have an information budget. So energy, information, materials. At a global scale, a cell has a budget, and we're basically using it to do what we want. And so redirecting it on that scale obviously impacts greater scales at tissues and others. I don't work in mammalian systems, but I would have to guess at a tissue level, you have yet another differentiation now that you have to deal with.
If I can follow up on what Ross said. So I've heard this idea many times that we are not building synthetic circuits to be exactly the same as the cell works. And I guess my question is, what is the difference and why are we not doing that? Someone who does experiments will be better qualified than me to understand this.
So Anya, is your question why we can't do what the cell does essentially?
Yeah.
Well, on the one hand, I play the devil's advocate here. I think the cell today does what it does after how many years of evolution, right? And been trying to optimize things and have seen favorable and unfavorable environments, and that's optimized the way it works. And we are trying to do that in the lab in 1 year. So I think to some extent, we are trying to shorten the timescales here by a lot.
Yeah, sure. Sorry.
No, go ahead.
Sure, but I guess are there fundamental ways in which the way we build synthetic circuits is very different than the way that the cell does things, and like what are the big missing pieces, I guess? So for example, one of the things is we haven't I think we haven't been able to engineer phosphorylation. So just and the cell makes abundant use of that to act on really fast timescales. So that's an example. But like, what are others? And do we think we'll catch up to them? I guess.
Can I follow up on that question, because that's a really interesting point you make there, Anya. So why replicate phosphorylation when you know the cell does it perfectly? Why not just try to hijack the system already?
I think some people have tried.
So has it worked? What hasn't worked?
I'm not a specialist in the area. Ross, any thoughts?
Yeah, I think if you're talking about these complicated signaling networks, the challenge is the off-target interactions, right? And so the cell has like evolved to have these complicated signaling networks where you can have multiple inputs come in and then provide diverse outputs based on the combination and timing of those inputs. And when we're trying to design systems, we're just going like one input, one output often. At least that's what we're starting with. And in a phosphorylation network, if you have any crosstalk within the context of that kind of system, then it's not going to work very well. And so one thing that we've done, like when I was working with Domitilla, was taking phosphorylation systems from other organisms that shouldn't have any crosstalk in a human cell. But that means that I can't necessarily with that interface with the signaling network in a mammalian cell, which you might want to do. Your engineering goal might be to control the signaling in the human cell, right? And so in that case, how do you design it in a robust way, I think is still like a big challenge.
Yeah, I like those explanation. That's wonderful. So also, Domitilla mentioned earlier, interesting point, evolution basically took, many years to optimize whatever system just by mutation, the random mutation. and then selection. And then we are trying to make something within PhD period or whatever postdoc period, and that's the problem, of course. But another problem, we currently have is, typically we use plasmid, high copy plasmid, and in essence, we have many genes in the bacteria or organism, and that not necessarily giving a lot of burden, but because we're using high copy plasmid to build a circuit, we start to see the burden. So, the many people also try to use the genome integration or single copy plasmid to build the circuit, but at the same time, that does not give a high signal to see. So we probably need to think a little bit differently when it comes to building circuit that could be very tightly controlled until we need to activate whatever circuit, or we need to use probably smaller number of copies. In that way, minimize the whatever burden effect. In that way, we could kind of mimic how biology evolved over the many years of many years.
Shugunshi, you have a comment? Yes.
Yes. So I'm just thinking one simple thing is here, and a lot of people say, for the genetic disease is genetics lost the gun, and the environment pulls the trigger. So most of this genetic disease is not the single genes, and it's complex. So then here, I just think about a lot of people. We are talking about the topic of today's topic. It's how the engineer can do at the same. You can sense that changes, which will cause the problems. So then we go back to the system, have the target. So then either to reverse that or make it beta. So is there any way we can make these things happen? Sense the environment which cause the changes or cause the problems. At the same time, we sense that, so then we respond to that immediately, and either to reverse that or make the beta.
So here I'll pick up back on a comment was made before, that we don't have generalizable rules that are applicable across different setups, different cell strains, different cell types, different applications. So the question is, if we restrict to that one cell type, do we believe that there are rules that one can use for that specific setup, or we don't believe that there are those rules that we can use?
Yes, we have.
Can you give examples?
So one example I already gave, that is the, Chris Boyce's science paper, and also your paper, regarding host interaction or retroactivity. That is, applicable. Retroactivity might be applicable to many different bacteria, because that is a fundamental resource, and not necessarily resource in a competition, but some other host circuit interaction. But the finding that from the individual cell or species, also discovering the parameter range might be tricky, because we need to understand those kind of host physiology and other things to manipulate and discover that kind of principle. But I believe there should be something because otherwise CRISPR never create that complex genetic circuit with the multiple different predictable design rules and then apply that onto the system.
Could you clarify the question? Is the question, do we believe that there would be rules that extend across species?
Yeah, so my question is more about, is there any hope to give design principles for creating predictable genetic circuits in general, or that's going to be problem specific? Every time I have this specific application with that specific problem to solve, with that specific cell, then I have a set of rules. But that's always going to be highly specific and there is no generalization ability.
I mean, it's an opinion, but my opinion or feeling is the answer is yes, we should be able to come up with a common rubric for how we proceed to engineer something. What kind of information we should know and what's critical. I'd say, what gives me hope that this should be engineerable across all scales is that every cell, regardless of what it is, has the same exact central metabolism, a very common core to it, and very common biological rules about interactions. We all use the same, more or less, amino acids, everything else. So from one level, the parts are all the same.
Somebody else before also mentions another aspect, which was interesting, that historically in the field, people have been designing circuits with the circuit-centric view. So just assuming that there is nothing around it and it's just the circuit, but I'm not sure if it was Ross or somebody else that mentioned that maybe we should change the angle in which we design things by accounting for the cell host to begin with. And that may change from the onset, the way we design things.
Well, one thing I've been thinking about with respect to what Mitch is saying here is that, and what Domitil was prompting before, how much do we need to know about the cell? How do these different contexts work? I think that some of the general design principles of how we model circuits, how things like feedback work are portable. Like we know generally how like an activator works in a plant, in a human cell, in a yeast, whatever. And there's the constraints of the cell, but maybe instead of, or one version of this is to make systems that are inherently robust, but those systems will still have their places where they could be fragile or certain parameters that are important that could still be bad. But we could still take advantage of that by saying, kind of going with the thing that biology allows us to do that other engineering disciplines don't necessarily, which is that you can create libraries and you can evolve things. So on one hand, there's been a lot of work making libraries and sort of screening for proteins or genes or whatever it is that have certain behaviors. And on the other side of this, there's the circuit design. And in some cases, those have merged. But if you're just trying to get, if we generally know that like we want some behavior, and we know that with some kind of feedback regulation and with a reasonable model of what this genes in the circuit are supposed to do, that it can work. But we don't know how it'll work in a specific context, then sort of building sort of library variants of some circuit that you're designing, but with an architecture that's robust, would make it more likely that you're going to be able to make something that in that context will work. I don't know if that works in all cases. Certainly some organisms are more, you're able to screen larger libraries than in other ones. And sometimes whatever you're trying to do is not necessarily going to be amenable to these types of situations. And how you measure success might also not work with respect to like a library type approach. But I think it's something that we can start to think about is how to bring those sorts of approaches to these types of designs so that you can get to a robust design that works in a specific context more quickly.
Thank you, Ross. I think Wilson wanted to say something. Yeah.
Yeah, sorry, I joined in late, so I don't know my what I said has been covered before. But I guess I start thinking a lot about design specifications. what, you know, so what are we designing for? Like, I think Ross was mentioning, I think, right, what context are we designing for, right? I mean, you know, if the environment is in, let's say it's dirty or whatever, it's like missing a lot of nutrients, a lot of competition. So we need to know about the resources available to the cell to begin with. Then we start thinking about whether it will impact our circuits or not. So I feel like a lot of the time we're designing things in vacuum, right? Without really thinking about what is it going to be deployed or used for. So I think that's the main thing, right? The understanding the design specification, then we can start really addressing how universal those design rules apply. And that could be very difficult, right? The chicken and egg question. Sometimes it's like, well, I don't know what your circuit can be, you know, your cells can be used for, so I don't really know, right? What environment it's going to be applied into. Is it in the soil? Is it in a gut micro, right? And those could have a very different challenge. So that's the thing I spend a lot of time thinking about these days. When I design circuits, I really try to kind of box in like what my specification is, right? I need to let it work for 1 week or 1 month, right? That has the different requirement on the robustness of the circuit as well, right? You might need to deploy a different types of circuit. Yeah, so that's what I'm thinking these days.
So thank you, Wilson. So putting together what Wilson and Ross said, so it seems that on the one hand, we want to have a better way of defining the conditions under which the circuit is supposed to work. And on the other hand, being able to develop architectures that are robust to uncertainty within those specifications, right? So that on the one hand, we try to achieve robustness, but on the other hand, we can't hope to be robust into everything, right, that exists. Okay. So, and regarding to that, oh, go ahead, sorry.
Yeah, I wanted to somehow try to maybe ask another question around the question that you asked about this, if we can design based on some design principles a circuit and then put it, for example, in a mammalian cell, and if we have a criteria that it works. So we know there is some problems with context dependency and then resources, energetics, etc. But the cells also have some mechanisms to stay stable, although there are some fluctuations in the environment. So they do their job, they're robust. So can we somehow try to use that perspective of robustness that the cells have and try to define a criteria for our circuits that can be integrated into different type of cells? But when you actually meet those criteria, you're somehow sure that your circuit going to work ideally.
So do we then believe that, picking up on this, on what Ross said before about architecture that is robust, so do we believe that there are rules or principles to create architectures that are robust to certain uncertainties or perturbations, either in natural systems or in SynBio?
Well, what I'm really interested in is to know how the cells stay robust, and then if we can learn something about the criteria that their circuits have, and then we can also implement it in our systems, where that's a possibility or not. That would be interesting to see.
Right. So like, how does a cell implement a feedback circuit, or how does a cell implement a feed-forward circuit? And sometimes those are simple, and sometimes it's layered. Like some of these signaling networks have multiple layers of feedback built into them. And is that necessary? Like they evolved to do that for some reason. Is it because they just need that type of control over their signaling networks? Or is it easy for the cell to do for some reason and not easy for us to do?
That's true.
So, the word robustness has been used a lot. And my understanding is that robustness is an emergent property. An emergent property is basically something we cannot predict without a better understanding of complexity and complexity science, which is really at the heart of all of our problems. And so I think defining robustness, like I think it was Wilson said, I want something to work for a week or a month, at least gives you some parameters to work on. But how emergent properties come about from first principles is, just not known yet.
I have a couple of thoughts. So inside the cell, what is it trying to be robust to? Like robustness is always with respect to something. And of course, there are actually architectures that we can use in synthetic biology to give more robustness to certain things. Like if you add feedback on top of something, it'll be robust to more things. But are we being robust to the correct things? Are we being when I build a circuit and I put it inside a cell, and I say I want it to work, I want it to detect some pollutant in water for the next week or whatever. What does that mean specifically in terms of robustness? What am I trying to be robust to? I feel like the discussions around robustness are always very vague and kind of like, what are we aiming for? What do we want to be robust to is very ill-defined.
Yeah, I think just picking up on that, right? Anytime we talk about robustness in control system design, we need to specify what property needs to be robust, right? The steady state value of such concentration needs to be robust, and to what perturbations. Changes in temperature, changes in nutrients, changes in concentration of such and such proteins. So I think that's what probably Wilson was saying, that types of specification, what needs to be robust and to what, right? You need to be specified better. But my question is, so what I'm not understanding is that people believe that there is no known understanding of how cells obtain robustness of specific properties to perturbations, right? So is that the belief here? So for example, we have no clue of how our body is so good at keeping that exact temperature. It doesn't matter if I have at -20 Celsius outside or it's boiling hot at 40 Celsius on the beach. Yes, my body is keeping my 36.5, right, body temperature. So we have no clue how that's done.
I would like to chime in there. I don't know about the body temperature, is a fascinating question, but we do know that cells are pretty good at compartmentalizing things. So they keep things in check by putting them aside in certain compartments within the cell. Going back to the phosphorylation and signaling, there's different implications when phosphorylation takes place at the cell membrane versus the cytosol versus the nucleus. Different chromosomes condense at different regions of the nucleus, so they sort of stick together and they don't mix up in a mush of DNA, swishing around. So do synthetic biologists think of compartmentalization in that way when they design their circuits?
Yeah, I also can add to what Tuba said, the environment. So for example, when you take out the cells from in vivo and put them in vitro, for example, and then you try to make an organoid, it is extremely less robust when you put it in compared to when you put it in this context. So part of this compensatory mechanism and feedbacks actually come from not only the cell, but from the environment or the niche that the cell is.
Also cancer cells too, right? When you think about them, when a cell becomes cancerous, it becomes robust, like it can take over under any condition, essentially. So I think I like Anya's point earlier that we need to define what robustness means in a context-dependent manner.
Wilson?
Yeah, I just want to add that, you asked about, if synthetic biology can do like spatial especially intracellular spatial control. I think we as a field, that's my view. Okay, we're molecular biology based, right? Three-dimensional structure engineering, it's incredibly difficult to do with genetic tools that we have, right? And that's both obviously at a subcellular level and also multicellular level, right? We haven't been very good at doing that. That's all. It'll be fun to think about, right? How do you control things at a subcellular level, spatially? But I feel like we don't even understand like subcellular organizations very well, right? I feel like the things I'm always trying to look at as a synthetic biologist, like what kind of knowledge that we learn, it's actionable. There are a lot of things that we learn, right? It's anything that I can actually use as an engineering clutch, right? That we can. I feel like spatial organization, like condensate, for example, and things like that. Those haven't been a point where I feel like it's a point. It's deployable yet. And there's a different thing. Sorry, I'm probably rambling. When we think about synthetic biology, I think about there are ways that you can monitor things. There's a lot of reporters you can use engineer to monitor events. Those reporters tend not to be very useful to control cell function because the dynamic range is too weak. You can have very sensitive reporter, things that change like 50%, threat and things like that. You can see an event, but you can't use those to control cell function. I find that to control cell function, you need a larger dynamic range. So it's kind of like what you said. You need to be able to control something that overpower the noise, the fluctuations in the cell in order for you to see the function that you want. So those tend to be kind of blunt instrument. That's what I feel like. That's like overexpress this really well up to a certain point and then overpower all the noise that the cell wants to do. And then it only does the things that you want the cell to do. So I find there are two different criteria. One we use to monitor cells, one that we use to control cells. These are two different sets of tools. Anyway, so yeah, it's a long story. I feel like it would be great to be able to compartment to control subcellular localization, but I don't think we're there yet.
I like the Wilson's point a lot. So I mean, we enjoy a lot from fluorescence protein regarding analysis of genetic circuit until now, but for the real world applications, we not necessarily require seeing those kind of reporters, because sometimes some applications only require very low level of expression. That we kind of learned that one by developing kill switch in my lab. I mean, we just need one cut in anywhere in the genome to kill the bacteria, not necessarily huge expression of whatever, Cas9 and, Cas9 protein. So that those kind of mindset change. I mean, of course, that paper already, the Jim Collins already kind of published one paper about one copy, plasmid system could work better. Still kind of very challenging to implement because we still rely on the fluorescence protein, and then we do not see, circuit behavior well in one copy system. But real world application, I believe that should be our next decade direction from the synthetic biology. Doesn't necessarily require high copy expression of the genetic circuit. That will solve lots of problems, including burden problem, those kind of host circuit interaction problem, I believe.
Yeah, I agree that a lot of these reporters, we can't detect them often until the protein is very highly expressed compared to maybe some endogenous genes are actually expressed. And the cell is producing tons and tons of transcription factors and can get away with gene regulation with many of those at very low levels, actually. by cooperating them together, among other things. And so maybe that's something we can learn from is, I don't know about making like tons and tons of unique transcription factors at low levels, but maybe designing circuits that can respond to lower levels of inputs so that we don't have to overexpress them as much, and then making reporters that are much more sensitive at the single cell level so that we can actually see whether it's working or not. I agree with that a lot.
I want to follow up on that point. When you design your circuits, do you design it with the intention of sort of be all approach, like you design one circuit to do like everything, the ring to rule them all? Because it sounds like, you can come up with a two-layered approach where you can, put something in there that's, low level, but then you can use an amplifier to amplify the system in a separate circuit or something. I don't know if it would work, but is it usually a single approach? You're trying to cram a lot of things into one design to achieve as much as possible. How does that work? Can somebody comment on that?
Yeah, I would just quickly say that you often don't have a lot of space to work with. And so, for example, if somebody's just trying to use a lentivirus to engineer cells, which is a really common approach, there's only so much that you can fit into the lentivirus. And so with that in mind, yeah, we're very limited. Now, CRISPR tools have advanced a lot, and there's other delivery methods that are being developed more that have higher capacity. And so I think we're getting towards the point where we can start to integrate more things. And I was also thinking about amplifiers. I think that's a great idea. Some way to just amplify some signal, even if it's just at the end to say, like, to detect whether what we tried to do worked. I've seen like projects or papers that people have published where they try to have little detectors for different parts of the circuit. I think Cello was a really good example of this, where they looked at how different nodes in their circuits were working to see if they could detect where problems were going wrong. But there's different ways you can imagine implementing detectors that would try to give you an idea of how each stage in your circuit would work. And something like an amplifier that you could choose what to look at would be a nice way to sort of take advantage of that without having to necessarily place a huge burden on the cell to give you some sort of signal from all of the nodes all at once as the circuit operates.
Yeah, to add one more point, when I was a postdoc, more than a decade ago, I used to believe complex circuit, the more complex, the better in terms of circuit, architecture or, the complexity. But now I believe opposite, the simpler, the better in terms of application down the road. Because, for example, you mentioned amplifier, that's a very interesting idea. People also try adding amplifier to kind of signal increase. But at the end of the day, you also need to consider trade-off between adding one more, amplifier or negative feedback. That addition would lead to more burden that kind of also may backfire. So we need to kind of consider the balance between adding, robust circuit module, but when you do that, we need to consider also you may break the circuit as well. So to me, application, the simpler, the better. That's my kind of philosophy change now.
So, so here is one point that seems to be coming up often, this notion of burden, right? That we are engineering circuits that are using what the cell provides, and therefore we are burdening the cell, and therefore we hit limitations. So we don't have that problem when we engineer electrical circuits, right? Because we engineer the power supplies. So is that an approach that may be feasible in SynBio, and is there anybody working on that?
That's a good point. I saw Eric hand. Do you? I don't know if you.
Yeah, I had a comment about what was said previously, but I can come back to it later.
Okay, yeah. Power supply. I guess, you know, I work in mammalian synbio. So, right, Matthias, so you know this more than I do, right? Their limitations is burden. That's a fundamental different burden than like engineering microbes. At least let's just talking about gene expression alone, right? So your work shocked me a little bit, I must say, a few years ago, right, when I learned about your work, because you know it shows that, right, there's a very unique thing that's being limited, that's different for different species or even different cell types, right? So again, we come back to the point that I don't even know what's limiting anymore. We're talking about gene expression, very simple thing that we do in SynBio, right? Have a strong promoter. In bacteria, it's ribosome, right? In mammalian cells, seems to be some kind of mediator factors at a transcription level, and that apparently is different in different cell types. I'm not surprised by after you pointed out, but I was surprised to see that at the beginning. So yeah, I oftentimes I don't even know what's limiting. It could be some rare, you know, at the end some metabolite, right? Whatever that I end up trying to control, right? Cofactors. So going back to the biology. So yeah, I think so. You know, recently, right? Like you've seen all these ADAR type of work, right? And they try to have this RNA that will get edited by ADAR. And it doesn't, I mean, it's amazing work, but it doesn't express well because it seems like ADAR activity is limited, right? So you have to overexpress ADAR. You could do that, but that's cancer, right? That's what cancer cells do. So so that you could do it, but it will cause disease. But yeah, there's an orthogonal ADAR, for example, that doesn't interact with the endogenous RNA. That only work with your RNA, that would be great. So that's a molecular invention. That's not a circuit level invention. That's how I see. So Toba, I just want to add back to your work. So, you know, you asked like amplifier, right? You know, there are different kinds of amplifier. You talk to like a circuit level synthetic biologist will build a positive feedback loop. If you talk to, let's say, a molecular, like directed evolution types of synthetic biologists, or what they don't call themselves synthetic biologists, they would go evolve a more potent, let's say, transcription activator. Right, that's probably what they would do. And then they would probably look at the structures of the thing and then see which one can bind to transcription factors better, polymerase. So they don't, that's an amplifier, but that's a different way of designing amplification. That's probably more space efficient, but of course, like Tae-Suk was saying, they have probably different kinds of trade-off, right? So I guess it's.
So yes, thank you. So I think the timer, so we have one more comment and perhaps we pick it back up after the break from Eric. So remember that, Eric, so we go back. So we have a 5-minute break until 2:35 p.m. And then we'll start with Eric comment and we'll go to topic area 2. Okay, so I think it's time to start the second topic area. So it's 2:36. So before starting, so Eric posted a very interesting question in the chat. So maybe Eric, you can just,
Thanks.
Yes.
Yeah, thanks. I don't want to take up too much time for topic 2. I just wanted to quickly, I was excited to hear everything that you guys were all talking about, the amplifiers. I think Ross had mentioned how cells get away with these low-level inputs. You know, really understanding, you know, selfishly from my angle, from a physical sciences approaches, looking at, you know, with cancer context, cell stiffness, we're really looking at how these phase transitions can really affect how a gene behaves, both good or bad, and how cell crowding, molecular crowding One of the claims is from an evolutionary standpoint, these, what is this metastatic state and how does this change, from a cell in its context of a multicellular component to becoming this unicellular cell state that wants to really affect its own proliferation. So I was just curious from those working in the field, and thanks, Ross, for posting that talk. I'll definitely take a look at it. Understanding, in terms of these guiding principles, can we really identify how sensitive genes are in terms of these physical parameters?
So Tuba, you had a comment back to that I saw in the chat.
I'm trying to find them. The phase separation. Yeah, my question was, when you design your circuits, do you think of the subcellular localization or the phase separation of these circuits from the rest? Because that could also feed into the resource competition issue. Like if you can somehow separate them physically, then you may not run into as much of an issue with, competition of resources. Something to think about.
I guess the paper that I posted here is maybe not so much like the physical properties of what you're bringing up, but I think it kind of borrows the concept of like phase transitions and imagining like the space of possible behaviors of like a circuit component in a way that those sorts of physical properties of the system are often imagined. And so I think that it could be like first bringing some of those like ideas of how to interpret or understand how like a circuit works into circuit analysis and design is useful. And then in terms of building circuits that take advantage of strictly the physical conditions themselves, I think it's really interesting. I think that we probably need to account for those kinds of things in the models, at least of endogenous genes, where clearly a lot of these higher order molecular assemblies are made. And in the circuits that we make, I think that they could probably help as well. So for example, in like the resource competition work that I did in Tom Attila's lab, we started to think about whether a model of phase separation of transcriptional resources would more accurately capture what was going on than a strictly like molecular binding model. And so I think that there's a lot of potential for that, especially in mammalian systems where you have tons of these types of multi-molecular assemblies that get made. And I guess the question will be how much more accurate are the predictions that are made by that compared to the standard molecular models that we make.
Thank you, Ross. Okay, so on that note, I'll switch officially to topic area 2. So here we change the gear a little bit. So we try to ask, if we have the application in mind, then what are the bottlenecks? Okay, so if we think of engineered cells usable, for example, for therapeutics or for basic discovery, to what extent issues of robustness, reliability, accuracy, and safety are on the way? What can we reuse or adapt from other engineering fields? So complex systems, or whatever complex means, there is a plethora of them, in engineering. So any other engineering field, such as aerospace, mechanical, automotive, and electrical engineering, had to overcome problems of reliability, repeatability, robustness, and accuracy in order to achieve tremendous goals, such as put the humans on the shuttle, send the rover to Mars, or qualify just a new vehicle for commercial use, which is actually really hard to do. And there is a common denominator in all of this. Control system design is one of them, okay? There are more. So a question here is, what are the common denominators? And can they be reused or extended, to address similar issues that we have in engineering biology? So, and with that, I'll open the table for discussion.
May I start?
Yes, sure.
So I already mentioned a couple of times earlier, I will say the key enemy of bioengineers, of course, mutation. Mutation always kind of making us to struggle, but at the same time, we could use evolution to generate more diverse circuit. In that way, we could benefit from that. But still, you know, at the end of the day, stability of the generic circuit, etc., always kind of suffer from mutation. And second issue regarding application, I believe, is biocontainment, because we're talking about unless you're talking about cell-free system, you probably also need to think about live cells, biocontainment, because at the end of the day, public do not like GMO, but we're generating lots of GMO. And then I would say one more thing I wanted to emphasize, not necessarily application, but in general, we as a researcher contribute to antibiotic resistant spread by dumping a lot of engineered bacteria and organisms that contain plasmid with antibiotic resistant gene, because we only kill bacteria and organisms by autoclaving or adding agent, but we dumping that into the sink, but that contain lots of plasmid that has the antibiotic resistant genes. So I would suggest we probably need to completely reconsider our practice of killing bacteria and then destroying even DNA. Otherwise, but people do not blame us. People blame, hospital and agricultural industry, because they use a lot of antibiotics to kill whatever pathogen for the cattle, pigs, and so on. But we also contribute a lot. So that is my 3 points we need to consider regarding synthetic biology moving forward.
Anybody wants to pick on those topics or anything else?
I talk a lot, but I'm if no one adds, I can add a little bit. But
Sure, go ahead.
So I work on cell and gene therapy, and the biggest problem I have is delivery of the gene itself, delivery large scale. That is the biggest bottleneck for us. I think some of I think Facebook was or Ross mentioning that we use lentivirus a lot, or mRNA, lipid nano AAV. Those have very limited size. And everything else about CRISPR, even with as powerful as CRISPR, I feel like the last couple of years, there's been a plateauing of number, like size of cargo we can deliver. It hovers around 5 kb right now. I haven't seen it past that. So I see that, of course, that's for human, for mammalian development, but that's a big problem.
So, so here then there are some points that have been touched. So one is the issue of mutations. We all know how disruptive that can be when you engineer a circuit, biocontainment, as it was mentioned before, and delivery of genes being a major bottleneck. So let's assume we have an ideal world where these 3 things have been solved. Do we have anything else on the road that impedes us to just engineer mammalian cells for therapeutics?
I have one thing here to think is, the biology is so difficult and so complex, and same since you worked in one even bacterial strain, it probably will not work in another bacterial strain. And the same thing actually happens in the different cells. The system works in the fibrosis, works very well, and when you apply that to to the stem cells or to the neuron cells, it does not work anymore. So this happens that maybe it's expression level and also epigenetics and also at the different, at the transcriptional and the translational and the post and the pre, all these modifications here. So my thinking here, one thing is, as a field, when we're talking about who are doing the basic discovery or who think about the therapeutic approaches, when we do the basic discovery, probably we do, in different organisms, in different systems. When we do the talking about the therapeutic things, whether we need a precise model to do that. Let's say it's neuron genetic disorder. Do we need to use the precise neuron genetic disorder model to investigate this system or these circuits in that in the neuron genetic models or not? Do we really need a very precise model, or we can use again, probably it's a question like robustness with precision or accuracy or sensitivity or specificity here. That's my thinking here. Thanks.
Thank you. That's a great comment. So what do we believe it's causing the fact that the same simple genetic circuit works in one way in fibroblasts, and as we reach iPSC state, it works differently? What's changing? So a few things were mentioned, like the epigenetics perhaps is an obvious one. What other things are on the way?
I think the tissue-specific things happens. Think about the liver cells, think about the neuron cells and brain cells, and they do have their different transcription or translational profiles there. It's very different. So then we engineer these things, those promoter activities, and sometimes, we use the endogenous promoter, sometimes we use, external promoters, like from wireless or from other organisms. So these things promote activities in different cells also have the rules here. That's what I'm thinking.
So what would be the design objective there? Then design a promoter such that its activity is preserved across tissue types, cell types. So what's the sense, and is that possible at all?
I guess like when I think about these cell types, so like here we're doing differentiation, or I'm trying to understand how cells develop. And for example, TM works on looking at how individual cells will make a decision based on their neighbors and on their own intrinsic state. And that's kind of at the top of a hierarchy of differentiation. And then we look also in our lab at how cells differentiate through a complicated series of events down to T cells. And so how does a gene, like, how can we predict whether it acts one way or another in one context or another? I think a lot of it comes down to the transcriptional environment that surrounds that. Probably, like, to me, the three most important things would probably be the transcriptional environment. So that includes the epigenetic state and the confluence of all transcription factors and transactivators that could be affecting the transcription of the gene. Then at the RNA level, the thing that probably changes the most would be, for example, microRNAs. This doesn't have quite the analog in bacteria, but I think maybe sRNAs in bacteria would be like a good analog in that environment that might be different between different bacterial organisms or bacteria in different states. And then at the protein level, yeah, if there's different combinations of kinases or proteases or whatever it is, that will affect in some way the protein stability or function. So is it sufficient to just sequence the cell and see what is expressed and then try to make a model that takes those and then predicts what the expression level or activity of a gene will be? I don't know. I think that it so far hasn't quite worked to use like motifs to predict gene expression patterns super well. But maybe that's just because we haven't had enough iterations on that and haven't been able to like study the cell in enough different environments in detail to where you could get the data to be able to make sufficient predictions. So maybe we can get there by doing this kinds of analysis, but we just haven't gotten to the point where we have enough to do it yet.
So in that sense, what's the trade-off between becoming really good at predicting the behavior of everything in the cell versus creating genetic circuits that somewhat keep the same functionality despite you don't know those environmental factors? So where do we draw the line?
I believe that's application specific. So I mean, I like the Ross point, but I'm a bacteria person, and then I can give my experience. We actually try to develop small RNA that are applicable to all bacterial system, including gram-positive and negative. And we successfully published one paper just using whatever simple linear regression model, or people call it machine learning, but we only have 200 whatever, different circuits, so we don't call the machine learning. But at the end of the day, what we found is that work actually. We found one simple mathematical model applicable to 3 different strains, but different condition require, because the small RNA also require some protein called HFQ protein. That is sometimes require, sometimes not require. So one simple mathematical model, I'm surprised because one simple mathematical model basically explain all the predictable behavior of small RNA repression, because I didn't believe that possible. But if you're talking about expanding that to other bacteria, I don't believe that will work, because there are too many unknown in another bacteria, because we only use Bacillus and E. coli, and that's well studied. But if you're talking about non-model organism, we even do not have enough tools to engineer the bacteria, also high throughput, transformation or some other tools. So we cannot generate 200 different variants very easily with that non-model organism. So current bottleneck is basically, let's say we wanted to use one bacteria, but we need to develop tools, and then to do so, it takes minimum 5 years based on my experience. So that is one bottleneck. But there are many useful bacteria that basically we can use other than E. coli and yeast and some model organism.
Thank you, Taisok. Tuba, I see you had a very interesting question in the chat. Would you like to ask that to the group?
Sure. Yeah, so I'm curious because, we're talking about what can we borrow from other engineering fields and what they had to go through in terms of the hurdles. But my question is, like, to what extent did other engineering fields have to replicate the natural world exactly as they were trying to mimic it? Because, I mean, a plane is not a bird, but they both fly. We learned from the bird to make planes to fly, and the goal is that they both fly. So the plane doesn't have to exactly look like a bird or act like a bird. So my question is, what's so different about the approaches in biological engineering and synthetic biology, such that, what I'm hearing from all the discussions is that there is an effort of replicating things perfectly, maybe without thinking the bigger question of what we're trying to do first. Am I reading it correctly? I don't know. That's what I hear. Like, are we trying to, making things perfect the enemy of the good? Like, should we just focus on what we're trying to achieve and pick the right tools to do it without having to exactly replicate? the natural system. I don't know if somebody could just touch on that.
So I think, yeah, so we have a few. Wilson, you want to?
Oh, oh God, I have a lot of thoughts on this. I teach, so I'll shut up first, unless somebody wants to jump in.
I think also Taisaka had something to say. That's
No, go ahead. I have a lot of thinking because, my first slide for my presentation always comparing bird and airplane, but I want to hear from Wilson.
Okay, let me see how much time we have. Okay, I think I tell my students, I always tell my bioengineering students that they are crazy because engineering life is much, I like to say it's much more difficult than other engineering disciplines because it's so messy. The reason why we chose to do it is because we are trying to, at least for me, right? We're trying to clean up the environment. We're trying to save people's lives. And those are biological, right? We have to interface biology. We have to send something into our body that will fix whatever ails us. And, we can, well, we send machines in, right? We send knives and, radiation, we send machine into people, and that does something. But ultimately, there's something that we realize that can't fix unless we send biological agent in there. And that's why we have to start engineering the cells. So I agree, there are things that we use are not perfectly used for that purpose. Like, for example, CRISPR is a big thing. CRISPR is a bacterial enzyme, right? We all know that, bacteria use to fight infection, but we don't use it that way, right? We use it to edit genome, right? This is a completely different use, and it's not perfect at all. We're not trying to replicate what CRISPR is doing in its natural context, right? It's a completely orthogonal thing that we are using for. So I think that's a really nice example where we use whatever tools it suits us, right? And that's not what is intended application, right? But I think we try so hard because we want to develop a new heart. People trying to make artificial heart, artificial pancreas, right? And with mechanical, electrical, but as far as I know, that doesn't work interface with biology well. Our body doesn't like it. So we try to engineer a real heart tissue instead, and that's why we try to replicate biology that way. So that's my thoughts.
Yeah, thank you, Wilson. You covered what I want to cover. So, as I said, I use my slide. My first slide always talking about airplane versus birds. The reason is, our ancestors dream of flying in the sky, but instead of creating birds, our ancestors created airplanes by analyzing analyzing birds, and then design wings and propeller and assemble them together. I believe synthetic biology, last 20 years doing exactly the same thing. Instead of creating completely new creature, we analyze existing cells, and then we design synthetic DNA and assemble them together to make engineered system that could, make biofuel, biochemicals, materials, even diagnostic, pharmaceutical, whatever, the therapeutic application. And of course, that is the synthetic biology to me. And Wilson, point out very important point, because for example, the computer very robust, electronics very robust, but we cannot use electronics to sense and actuate to cure disease in response to disease marker in the human body. However, biological system, including probiotic, some microbiota, could respond to the disease and then generating some protein or chemical that basically minimize whatever impact of disease we currently have. So that's why I'm so excited with the biological system, despite all the messy problems in biology. And actually, I asked one question to Kristalla Preder and Jay Kisling in metabolic engineering, perspective, because I still believe biology. Sometimes I fear art, and then I asked the question to them, saying, why we still have, why we still don't have forward engineering, completely forward engineering, predictably designed metabolic pathway. And then why I still feel like we are still doing art in metabolic engineering, even though metabolic engineering basically more than 30 years. And their answer was, biology is messy, but we need to harness those messiness to address some practical problem, and that is a metabolic engineering. Same thing for the synthetic biology or biomedical engineering. We don't necessarily need to solve all the biological messiness problem. We just need to harness some of that for the practical application, I believe.
Thank you. So I just want to comment back to the comment that Tuba made. So it was easier to hijack gravity than hijack biology. So I just want to make two comments to that. So the first is that to hijack a gravity, you just need to know that F is equal to ma. And the fact that, so the way the airplane wings are designed is that the velocity of the air, right, in the upper part is different from the velocity in the lower part, and that creates a pressure difference that lifts the airplane up. So that's essentially the design principle. So there is one design principle that is very well known. And the other thing is that even with that, you need robustness, right? So we have heard about, birds flying into the engines, right, of airplanes. So, but airplanes can, and that happens often, that's sad, but that happens often. But yet airplanes can still work with just one engine working. And because there is the redundancy and there is a lot of robustness in the control circuitry that maintains the balance, right, and the lift of the airplane. So I think it's a conjunction of very well-defined physical principles, so physics, and very well-established mechanisms for robustness to known perturbations, right? So that's the way I would view that.
And I want to add to that too. You think about the sky, it's not that unpredictable environment. You kind of know, you can with these days with the weather tracking, you kind of know what's up there in the sky, right? It wouldn't have like dinosaurs flying out of nowhere. I think if it's like really messy, have all kinds of like things that you don't know that's flying up in the sky, we wouldn't be able to fly the airplane. There's essentially nothing up there, right? That's why it's robust and safe. Right, that's why an airplane is safe, because you don't have crazy driver up in the sky, right? Who's like checking text on her, right? You have very well controlled environment up there, right? So another thing I also want to add up, this airplane and bird analogy is used a lot, but I would say the bird, like our airplane doesn't hover like birds can, right? It's amazing, it can fly 5,000 miles, right? In a few hours, but it can't stop on a branch on a dime, right? It can't just jump off and fly, and then stop on the branch. Like we can't do that. Our airplanes can't do that, right? And of course, birds can haul like tons and tons of stuff across oceans, right? So they're quite different at the end of the day. Yes, they both fly, but the use are very different. I would imagine, if we want something to hover like a bird, we're gonna end up engineering something really like a bird, right? I think we just.
So to pick up on that, I think you said something really, key, right? You said, well, for example, for the airplane, or even, right, the shuttle that flies into space, right? There isn't so much uncertainty around it. So there are perturbations in the environment, but there's no way close to the level of messiness and uncertainty that we have to deal when we engineer a circuit, right, in the cell. So then here the question is, are then the tools that we have in engineering for simulation, prediction of behavior, and engineering robustness to those few uncertainties not adequate for engineering biology, right?
I mentioned in the chat, but redundancy, and we also mentioned redundancy, but redundancy would be one way to solve the problem, because redundancy sort of, address the mutation problem. So let's say one circuit die, but we can have another redundant circuit that kind of back up. And that is actually the way biological system evolved for many, many, years, because some critical function, they have some backup, redundant copy somewhere in the genome. And then we could implement that one in biological system. Engineered biological system, I would say.
Yeah, so to follow up on that, I think the reason why people probably don't do that is because of the burden issues that we were talking about earlier, that if you had many copies of the same circuit or same parts of the circuit, you would run into problems with resource limitations inside the cell. Maybe that's why.
Yeah, but I can tell you, I already mentioned earlier, that's why we need to probably, away from the high copy plasmid when it comes to building circuit. I had a recent experience. We basically put 4 different Cas9, copy with the different promoters into the genome. And we did not see any burden problem in terms of expression of whatever reporters in terms of growth. Because I mean, you can imagine, as you can imagine, we have many, many genes in the genome, but that is not necessarily expressed all the time. But just having 4 copy in the genome, it doesn't necessarily giving a lot of burden. And also, if you have a tight expression system that doesn't express until you need that function, and the cell doesn't experience any burden issue because we only have 4 copy with whatever distant expression level. And then for my application, basically this kill switch, you don't need multiple copy of the Cas9 protein, because we only need a few to cut the genome at the end of the day, because genome is just 1 copy or 4 copy, depending on which, growth phase you kind of having. I'm talking about E. coli. So, I mean, that might be the reason we don't see any burden. And then I highly recommend we kind of transfer from high copy, system to smaller copy or 1 copy, integrate everything in the genome. Technical challenge would be, of course, the high throughput, because plasmid is so convenient.
So the other thing that was mentioned before, I think by Wilson and probably Ross, is that there is also bottleneck to the length, right, of your synthetic DNA that you can put in the chromosome of a mammalian cell in this case. So if you do lentivirus, you are bounded by 9 kb more or less, and maybe piggyback you can do larger, but still there is a limitation there. So even then, so there is a question, right? So burden may be limited, but maybe there is a practical length issue. So I also saw that Tuba had another interesting question. So Tuba, you want to go with that?
Yeah, sort of following up on what you said, Domitia, there are bacterial artificial chromosomes that people also use that you can deliver up to hundreds of kbs of payloads with, within endogenous contexts and copy numbers. So is this a tool that the synthetic biology field uses?
Very rare. I know Pam Silver uses them, or engineering human artificial chromosome. I never tried. So I use a lot. I mean, of course, you use back, you have to use cell line that you can engineer. If you try to do gene delivery, that's, I don't think you, yeah, I never try to think about how to deliver back, directly into people. But I would imagine very, very inefficient.
Yeah, I think that delivery efficiency is a big problem because like even piggyback, you can have a huge capacity in theory, but how do you deliver it to the cells efficiently? Most cells are not that easy to transfect, especially primary cells or differentiating cells or whatever it is. Lentivirus tends to be a bit better in this regard, and just why people use it, but even lentivirus can have issues with different cell types. So I think the delivery challenge, like getting around that, will help us out a lot. How do we deliver large cargoes where we could implement more sophisticated, redundant types of networks?
What about exosomal delivery or liposomal delivery? I don't know how you deliver lentiviruses, so that may be a silly question, but.
Yeah, I don't know. I think there's even, yeah, I don't know about exosome, right? But like these liposome nano, there's like size, right? There's only so much you can pack in at the same time. So hundreds of kb is huge. I don't know if you can even package them, but yeah, exosome, I don't know. Microsome, I don't know how that works. Yeah, that's interesting. Sometimes I think about if you can have small pieces and then somehow assemble inside the cell instead of having one gigantic piece trying to shove them in there. But I don't know how to get all the pieces in and assemble them, like using kind of like what yeast, right? People do that in yeast, right? They deliver small pieces and have them do homologous recombination inside yeast, right? Am I understanding how people make yak?
So Wilson, you mentioned before that Pam Silver used the bacterial artificial chromosome to introduce a very large payload.
I think I think Pam Silver and yeah, I think they're trying.
Okay.
With John Glass, I think they are engineering human artificial chromosome. So it will be replicable, right? It will be heritable. So the extra set of chromosomes that's heritable. I think there are people trying to do that. But I don't know the status of that kind of work.
Okay, so going back, okay, so here we have a question from Jumont, right? What can we learn from current historical successful or unsuccessful therapeutics like CAR-T, gene therapy, new biologics? So any insights on that?
I mean, besides the fact that it takes a really, really, really long time.
I think on the delivery side, what's interesting is that the lentivirus delivery has been relatively safe. I think that the old gene therapies with retroviruses didn't work very well due to safety issues, but lentiviruses seem to be surprisingly safe from CAR T cells, from what I understand, although I don't do that myself. But what has been interesting is that piggyback and other transposons have had some issues in therapies that have been developed with them. And seems like it's related to the copy number issue where you can get you can get, if it's a good thing that you want, very high copy number integration with piggyback, but it can also be a problem because you could have a higher chance of insertional mutagenesis or some other issue. So I think that at least on like the delivery side, I think we can learn that certain vectors are sort of safer to deal with. They limit their integration frequency into the cells better. And as far as like creating a useful product that is less likely to perturb the cell, something that's sort of self-limiting in that way is probably going to be better as like a foundational element to work with. Or if other systems can be engineered to be more like that, where it's more self-limiting, more constrained to where it goes. I think, again, with like improvements in CRISPR editing efficiencies and new ways of delivering donors for engineering cells with CRISPR, that we'll get to the point where we can very specifically control where we put genes, and that should be even better, even cleaner, and even more safe in that regard. But I think that on the side of genetic engineering and just like general synbio, like how we deliver genes matters a lot. And whether it's in a plasmid or whether it's in the genome or where it is in the genome, we know that it affects how the system behaves. And so getting a better understanding of why is probably useful. And maybe the CAR-T example gives a simple illustration of like the perturbations to the cell behavior that can happen depending on how you do it. But maybe there's more to learn there too about what is the most effective at controlling different aspects of the cell behavior too.
Can I ask a related question? It's interesting that this delivery issue has come up. From my understanding, lentiviruses are used in humans because of safety. But a lot of synthetic biology is not done in humans. It's done at the cell level, maybe organoids, right? So why, you're not limited to lentiviruses to infect a cell with a large construct. Like, why focus on, I understand the ultimate goal of treatment in humans, but like we're not there yet. And maybe by then we will have the tools to be able to deliver large cargoes in different ways in humans. But why are you guys limiting yourselves to just using lentiviruses if what you're focusing on is bacteria or cell culture or maybe organoids at most? The tissue complexity wise.
In fact, oh, go ahead.
Oh, Wilson, no, go ahead, please.
No, I would say this is what NIH want us to do, right? To have the applications, right? And so, right, that's why I move into human. I agree. I would love if we have the space to explore with cell lines and model systems, and we can learn about the design principle without worrying about where it will lead us to in terms of application, right? It will be fun to do that. But that needs resources where we don't have to think about immediate human health application. But yeah, we could. But even then, right? Even with cell lines, right? It's still not that easy to deliver large DNA payload, especially when the efficiency is low. And let's say if you deliver efficiency is like 0.1%, that means that you gotta have a large clone, right? Large number of cells to begin with and select for clones and then it grows up. Those takes time. We do that for stem cells, right? And we try to do that through editing, and those are, takes a long time to grow them up and then do the experiment. If you have a large efficiency, it will be much faster.
Yeah. Yes, and on top of that, right, with low efficiency, you have low molecular counts. And when you have low molecular counts, everything is stochastic. And so it's very difficult then to predict, right, the mean behavior of the system.
Yeah, and if you're trying to engineer like a dynamic system, so for example, cells in the middle of a differentiation process or cells that are in the middle of some other kind of transitional process, you can't necessarily like engineer those cells and then select them unless, again, you have just an enormous number of cells that you start with. And then maybe you could sort some of those cells out that are engineered in the way you want. Lentiviruses tend to be more efficient than a lot of other ways of doing things. So you might constrain yourself to something like that in such a situation. To do a CRISPR editing of like a CD34 blood progenitor cell is getting better, but that's still really hard. And it's hard to keep those cells as stem cells when you do it. So yeah, I think the tools are getting better to be able to do this kind of thing, but it's still very constrained in terms of stability of cell types. And even in PSCs, like there can be stability issues when you try to engineer them, even though they can be expanded stably in many cases. So yeah, the stability of the cells is an issue. The scale of how many cells you need to parse through is an issue. And yeah, the bigger the cargo, often the less efficient you're going to be at doing that in the first place.
So,
Oh,
sorry, German, go ahead.
I just wanted to add one more thing. I cannot speak for mammalian engineers because I just started that at mammalian fair last year, but I can speak for the bacterial community. Actually also my lab also I wanted to develop some generalizable tools that could be applicable to all the bacteria, especially also I'm thinking about microbiome engineering would be the next thing in the next decade. And then in terms of transformation and the delivery of the gene or circuit into the microbiota, we found liposome delivery would be the best because that doesn't necessarily require any special tools other than liposome can sort of fuse with the membrane and then fusing the cargo basically go into the cell, but still efficiency issue there, generally not efficient but still workable in a sense. And then, but I'm not sure how liposome working in human cell and other eukaryotic system because I never tried that one in my cell. But I guess there are some tools we could improve to engineer all the microbiota without having any transformation issue, because without transformation it's impossible to engineer those kind of microbiota. But we have some tool developing, and then people also develop some tools. Also another delivery tool would be probiotic. You could use the live cell to deliver whatever therapeutics to gut or even skin by using skin microorganism.
Okay, thank you. So just as an interim summary, I've heard that bottlenecks to practical application and include, of course, we have talked a lot about this delivery issue, which is a purely biological problem. We have heard about the messiness of biology be no way comparable to the uncertainty that we have when we engineer systems. And then on the other side of the spectrum, I've heard about tools. I've heard about machine learning for prediction. I've heard about feedback control, feed-forward control. So the question is, if we had delivery issue ruled out, is robustness the only issue that we have? And yes or no? And how do those tools that were mentioned before could be used to address that or are insufficient?
I actually think that if we do understand our environment well, both inside the cell and outside the cell, I think we could engineer robust systems. I think we do know quite a bit. Like, it is the unknown that mess things up, right? Of course, I think Damatilla, you mentioned like how much do we need to know, right? That I don't know. That's a philosophical. I think apparently not enough. I think the diversity of the chemistry in the process that surprised me is just overwhelming sometimes. But I think we're getting there, right? The information that's coming out, it's really fast. But yeah, I feel we are making ground, you know. For example, I think a lot about tumor, right? And then there's like, we're now not just about the genetics, but about the tumor microenvironment, the metabolism within it, right? Now we start getting into, you know, the food, the energy source, and all these things, and then we can start factoring them in. I am hopeful that we're able to develop more and more robust based on these information, right? I think a lot about, you know, when we like space launch, right? If you know that there's cloudy, we don't launch, right? The weather's not good, you don't launch. So like this information is extremely valuable to the success, right? So I think we're getting there when I'm not sure.
And so how about tools? So we have heard about some tools, but do we think there is the need of a fundamental, you know, approach or a paradigm shift in the tools we are thinking of? Are we just trying to borrow tools as they are from other fields and feed them in while they just don't fit? Or there is a hope for recycling?
I do have questions for you for that. Like, how much from engineering principle, like, you know, about, I think someone mentioned about uncertainty, right? Right. How much, you know, let's say AI or these other maybe control theory, right? When there might be some theory even then, like how much uncertainty you can have in your system that you still can have some kind of robust system, right?
Yeah.
But the thing, of course, I don't know these theory, and I don't know how to implement or even test that in a biological system. So what's your take?
Okay, so that's a very interesting question. So there is a lot of work being done in what's called physics-informed machine learning, which is this idea of using machine learning to improve our models. So you add neural network or recurrent neural network models to your physical model so that you reduce the uncertainty around it. So it's viewed, at least in control system design, as a way to reduce the uncertainty so that you can make less conservative control designs. So I think it would be very interesting, right, how and whether we may be able to apply something like that, right, to SynBio, because I feel that, yeah, so we have the central dogma, we can write the Michaelis-Menten kinetics, right, for enzyme kinetics, but at some point, once we interact with the cell and with the environment, there's so much unknown, and can we write equations for everything? Probably not, right? So there is a limit there, and I know several people in the field are thinking of overcoming that limit by adding machine learning, yeah.
But you need like data, right, to feed into these models. The thing I always struggle is, what kind of data should we get?
I think that's a very interesting question, and how much data is sufficient. So on the one hand, if you constrain a machine learning model with knowledge from physics, you need less data in theory. So that's one hope. On the other hand, there is a question about if you need to determine an input-output relationship, what do you need to measure, right? And how many measurements? Is dynamic measurement, is static measurements? So I think that's a pretty open research area to me, yes. Okay.
Thanks.
Any other comments or questions from anybody, even the people that have been sitting silently?
So I've got one last comment. In my previous job, we were always designing things, for maintenance, right? You want to design with maintainability. That was one thing, because you don't want to take an airplane apart just to change the oil. Can we apply, or are we applying this in circuit design? Are we designing with robustness from the start? I don't know. I think we do the simple things right now. We just do the things that something works, and then we go back and try to add robustness. But that, to me, when it made sense, and it's, I don't know if it's a paradigm shift in genetic design.
Yeah, I think this ties back with something that was mentioned before by somebody else, that so far genetic circuit design has been based on designing that specific circuit and then worrying later about its interactions with unknown components in the cell, and then asking the question, oh, this isn't robust, so how can I fix it? What can I build around it? But yeah, so I think the moment that you say, I want to start to build the circuit from the start such that it is robust to end things, that would probably be a change of mindset, I think, in design with respect to what at least I have seen so far.
I think that just adds complexity.
Yes.
Yeah, I wish in the Domitillas in a new approach, or that I believe that is the right approach to do, but practically very challenging, because we are all busy, and then until my students showing promising data, I do not show any interest in that circuit, architecture, and then we start to worry about host interaction and burden and so on. And that is kind of reality, unfortunately. But I strongly believe, Domitila, you point out very important thing. We need to think from the, host level point of view when you start building something. Otherwise, we'll see a lot of problem at the end of the day. And of course, student could publish something, but real application, we need to think about all other factors.
Right. Yeah, I think this also ties back to something that was mentioned some times back, is that so the focus so far, even for, as you're saying, we are all busy and we try to get that cute circuit published that will get into nature, right? But then we worry later, oh, by the way, that's not quite robust, right? But yet you have shown something, right, really cool, right, that gets published well. And I think robustness, somehow, it's an issue that maybe we often try to put under the carpet because it's not such catchy, and it's just, right, a careful engineering job to try to figure out how to design things for robustness. But that's my view. Okay, so we have 5 minutes left. So do we have any last minute comment or question before we convene for making our summaries?
We should talk about later on this reward system, right? What did you get reward on, right? You don't get reward for being really, really good in engineering for being robust, right? You get building something barely works, but it's the first one out there, right? That's that's unfortunate. But I asked this for like a journal editor. It's like, how do we deal with this? And then the journal editors punted back to us, like, well, you guys criticize each other's paper that way, right? So we as a community probably have to change how we set the bar.
Right.
Well.
Yeah.
Right? We don't kill each other for improving the robustness from like 80% to 99%, right? We should reward them.
Yeah. Actually, I start to ask my student in the test circuit for 1 month by subculturing for 1 month, but that's probably too much because they need to come to lab every day for 1 month. But I start to ask, because I care about robustness a lot. But the problem, as you said, I mean, the old system, Nature, Cell Journal, not that much care about robust circuit. They wanted to see sensational or sexy circuit or application at the end of the day.
I guess it goes back to when earlier we're defining robustness. I mean, things can be robust in a set range of conditions, right? So if you can get a system to work reproducibly within the confines of certain parameters and show that it works consistently, I think that's good. I think when we think about robustness, we think we want to throw everything under the sun at it and it still holds up. But I think if you can find that range you want to work in, I think it goes to what you had mentioned earlier, Wilson. What's the goal of the engineering application? And if we can get it to work in that range, then I think we're onto something.
Also, I'm starting to realize the difference in the approach in the studies between like biologists and engineers. Like a month in the lab for us is like nothing for an experiment. I've done experiments for months at a time that didn't give me anything, and you just start over again. I've grown liters and liters of cells just to get one tiny amount of, protein that I could analyze on mass spec. I grew millions of fly embryos to get the right amount of, material that I needed for my experiment. So I think just thinking a little bit bigger and in a bigger context, and just not being afraid of maybe scaling up to get to the heart of what it is that you're trying to do is something I just want to put it out there.
Cool, thank you. So how are we doing on time?
So my 1 hour alarm is just about to go off, so I think that's perfect.
Okay. Okay, sounds good.
Seconds left.
So then shall we adjourn here unless anybody has any additional comments? And otherwise we'll go back and do the homework of writing the summaries and the slides. No? Okay. So, well, then let's adjourn the session and we hope to see all of you at the workshop on the 14th of December.
Yeah, I think we've got some closing slides. So I'd like to thank Domitilla and Tuba for leading this really robust discussion. And yeah, so we've got one more that's scheduled and another one they're still working on. So Eric and Marcella are going to lead the next one on the 6th of December, scaling up biocontrol methods to complex biological systems. So just a little branch of part of the discussion that we had today and before. So if you go to the next slide, our virtual meeting, the culmination of all these breakouts into the workshop. This is our annual meeting. We're doing it virtual this year, December 14th, and you can see the link that Holly put into the system, but you guys are successful at signing up for this one, so the main one is there. And we'll hear reviews from Josh, we'll hear reviews of Josh's work, and then the summary, Domitilla's work, and then the summary, and then Marcella's and the summary. So I think that's it. Yeah, thanks for joining us. And I've got a lot of homework. I think my favorite comment is Wilson's, the bioengineering students are crazy. So I got to put in the summary somehow.
Oh no, oh no. Yeah, I always say they didn't know what they got themselves into.
Yeah, okay, they didn't get them, yeah.
No, that's, yeah.
Okay, thanks again.
See you.
Bye, thank you.